Good morning. Welcome to the course Demystifying the Brain. This is the first lecture on the history of ideas of brain. You might be an excellent artist with long slender fingers and a hand that can produce delicate brush strokes. You might be an athlete with strong legs that can push you forward at a whopping 28 miles per hour. You might be a model or an actress with a face that can launch a whole naval fleet, to borrow a powerful metaphor from the English poet Christopher Marlowe. You are proud naturally about these physical attributes that makes you famous, but we rarely pause even for a moment to think about the real organ that lends these other visible organs their power. It is a hidden talent behind all our outward visible talents. It is the master organ that controls all other organs in the body, and that organ, ladies and gentlemen, is the brain. All of us have brains and we are generally quite happy about it, but here is the hitch. When someone says, I have a head, it is a statement of visible fact, but when someone says, I have a brain, it is most probably a bluff if you take a strictly scientific attitude, because you can see your head in the mirror, you can feel it in your hands, but you cannot see or feel your brain unless you get it scanned in an MRI machine, of course. But then you see something like this, not exactly something you would like to show your grandchildren. You do not feel your brain normally because brain is hidden, secured in the bony fortress of the cranium or the skull. You can feel the gurgling noises your gut makes when you are hungry. You can feel your heart pounding away when you are thinking sweet thoughts of love. But you can't feel your brain. Brain chooses to be that silent and mysterious controller behind the screen, that invisible orchestrator of all your life. This mysteriousness of the brain had intrigued people since the beginning of history, because brain did not suddenly show up yesterday. People had brains all along, although certain things that you read in history seriously question that belief. Let us rewind to the ancient world and see how people thought about the brain. Let us begin with ancient Egypt. There was a papyrus that dates back to 1700 BC, but probably based on earlier texts going back to 3000 BC, that describes the anatomy of the brain. It speaks of the meninges, the coverings of the brain, and the cerebrospinal fluid, a fluid medium in which the brain is suspended. Ancient Egyptians worshipped a lot of things, but the brain was not one of them. They thought that the heart is the seat of the soul and not the brain. Therefore, when they prepared their mummies, they scooped out the brains through the nostrils, but left the heart in place. Let us now move forward to ancient Greece. Greek philosopher Alcmaeon of Croton, who probably lived around the 5th century BC, had questioned the ancient idea that the heart is the seat of the soul and gave that place to the brain instead. But later, in the 4th century BC, more popular Greek thinkers like Aristotle held the traditional view that the heart is the seat of the soul. He even said that the purpose of the brain is to cool the blood pumped by the heart. The real truth is the exact opposite. The blood, among many other functions with respect to the brain, helps to cool the brain by carrying away the heat generated by the brain. But then Aristotle freely speculated about a lot of other things in nature. For example, he said the comets were tiny whiffs of gas burning in our atmosphere. He said that heavier objects fall faster than lighter objects. He thought that earthquakes are caused by the action of the wind on the earth. In these, as in many other things, he was dead wrong. Galen, the noted Greek physician who lived in the 2nd century BC, performed extensive anatomical studies of the brain. He captured his findings in a prodigious volume called On the Brain, in which he described the brains of a wide variety of animals, including cats, dogs, weasels, camels, lions, wolves, bears, mice. Apparently, he spared the insects. Look at this graphical description of the procedure to dissect an ox's brain in his book. When the brain is suitably prepared, you see the dura mater. Slice straight cuts on both sides of the midline down to the ventricles. You will observe a third ventricle between the two anterior ventricles with a fourth behind it. Although Galen knew a lot about the structure of the brain from his anatomical studies, When it comes to brain function, he held on to some old and wrong ideas. He thought that there are these animal spirits, the Greeks called them the pneumata, that animate the brain and are responsible for all its functions. Then came the Middle Ages. Not much has happened during the period, except that in the Islamic world, some scholars have kept the lamp of knowledge glowing. Al-Zahrawi, who lived in Islamic Iberia around the 10th century AD, was an eminent surgeon of his times who performed surgeries for head injuries, skull fractures, spinal injuries, hydrocephalus and so on. Here is a page from Al-Zahrawi's treatise on medical instruments. Let us fast forward to the Renaissance period. Renaissance was a time when Europe was breaking the shackles of the Dark Ages. Great strides were being made in the fields of literature, art, music, science, geography and commerce. Leonardo da Vinci, one of the key Renaissance figures who is famous for the Mona Lisa painting, also performed extensive anatomical studies on human cadavers. His objective was to study the human form thoroughly and use that knowledge to perfect his art. His studies of the skull and the brain have an immense scientific and artistic value. Another influential personality who had a lasting impact not only on the evolution of the brain science, but science in general, was the French philosopher René Descartes, who worked in the earlier part of 17th century. He helped to resolve the long-standing turf war between theology and science by stating that the world of matter with which science is preoccupied and the world of the spirit which is the domain of theology are fundamentally different, each governed by its own set of laws. Impelled by such a clean severance from the influence of theological explanations of brain and mind, thinkers began to seek more rigorous and evidence-based explanations of how brain works. By this time, the modern scientific revolution is fully underway. Galileo had already established the power of the experimental method. Newton had already worked out his mechanics, unraveling the wheels of the universe. From this point on, parallel developments in various branches of modern science have contributed to various aspects of brain science. Let us begin with the developments in anatomy. As we have seen before, there was a reasonably sophisticated understanding of the brain's anatomy that goes back to about 3 millennia, if not more. But that was only gross anatomy of the brain. People did not know much about how brain looked at the microscopic level, at the level of its cells. People learnt about the existence of cells when Robert Hooke made his first observations of the cell in a microscope. Normally, cells are like fluid-filled blobs disconnected from each other. But people thought that brain cells are different. They thought that brain cells form some sort of a gooey continuum in which fluid can flow from one cell to another. Such a tissue is called a syncytium. Part of the reason why they thought brain cells are different was because they were almost transparent and were not easily distinguishable in a microscope. But the scene has changed completely when Italian biologist Camillo Golgi had developed a way of staining or coloring brain cells so that they can appear as distinct entities in a microscope. Ramon E. Cajal, a Spanish pathologist, had performed a large number of microscopic studies of brain tissues and concluded that brain tissue is not a syncytium. Brain cells are discrete individual entities which he called the neurons, a name that resonates with the names of some of the fundamental particles like the electrons or protons. There is no continuous corridor that connects the neurons. Somehow signal to each other across the gap of the extracellular space that separates them. This new perspective of the neuron is called the neuron doctrine. Neurons make contact with each other at a special place called the synapse. At the synapse, the so-called presynaptic neuron has a flat button-like feature which comes in close apposition to a similar flat feature on the postsynaptic side. Cajal guessed that there might be a tiny gap between the two structures, and he guessed right. The gap was invisible to the microscopes of his day because it was only 20 nanometers wide, but confirmed subsequently by developments in microscopy. However, at the end of 19th century, when Cajal had proposed his neuron doctrine, it was not clear what exactly is the nature of the signal generated by the neuron and what gets transmitted from one neuron to another. The story of neural signaling begins in late 18th century. Earliest clues regarding the nature of the neural signaling came with the work of Luigi Galvani, an Italian physician, physicist and biologist who lived in the second part of the 18th century. In a serendipitous experiment, Galvani's assistant touched a dead frog's leg with a charged rod. The leg twitched as if it were alive. Galvani immediately appreciated the link between electricity and life in general and muscular action in particular. Galvani's work paved the way to the branch of biology called electrophysiology, which studies the phenomena related to electricity in biology, the brain being an important example. Another major milestone in the history of electrophysiology of the nervous system was the development of the electroencephalogram or the EEG. EEG basically consists of recording the voltages produced by the brain's electrical activity by placing electrodes on the scalp. Earliest work in this area began in the late 19th century when people recorded EEG from animals. The first EEG from humans was recorded in 1924 by German physiologist Hans Berger. But studying the brain using scalp electrodes is rather crude because it does not give any insight into how the electrical signals are generated by the neurons. A major step in solving this problem was taken by British physiologists Alan Hodgkin and Andrew Huxley, who performed experiments to understand the electrical properties of neuronal membrane. For this purpose, they record from the thick nerve fibers of a large animal like the giant squid. They describe the underlying physics of the neural membrane using mathematical equations. They unravel the electrical processes in the neuron that generate a sharp voltage spike called the action potential. These developments in electrophysiology explain how a neuron generates its electrical signals, but one question was still open. It was not clear what kind of signal goes from one neuron to another. For a long time, people thought that the signal is an electrical signal. It was believed that current from one neuron A directly flows into neuron B, since the brain was thought to be a syncytium. But the neuron doctrine posits that neurons are distinct individual entities. It was also proved, as we discussed before, that there is a clear gap that separates the two neurons meeting at a synapse. What then was the signal that jumps across the synaptic gap? Insights into the nature of the synaptic signal came with developments in the field of neuropharmacology. The effect of chemicals on the nervous system were known for millennia. There were neurotoxins like muscarine that were used by hunters. Arrows tipped with these chemicals could immobilize the prey by directly acting on their muscles. Intoxicants like alcohol with obvious effects on the brain were known since the beginning of history. The work of Paul Ehrlich in Germany and John Langley led to great insights into the action of chemicals or drug molecules on cells. Paul Ehrlich suggested that chemical compounds could be made that can selectively target a disease-causing organism. A toxin that can kill the organism can then be allowed to ride along with the selectivity agent. He called this arrangement a magic bullet. John Langley, on the other hand, suggested that this specific action of the drug molecule on a cell is enabled by the presence of a special substance on the cell membrane. He called these substances the receptor molecules or receptor substances. Subsequently, it was discovered that there are protein molecules present on the cell membrane that act as receptors for transducing the action of the drug molecules. But this work led to another fundamental question. What are the receptor molecules doing when they are not responding to the drug molecules injected by the experimenter or the patient? What is their normal function? The question led to the idea that the receptors on a neuron normally respond to the chemicals released by other neurons. That is, neurons communicate with each other by exchange of chemicals. This is a powerful idea, but it needs to be proven. That proof came with a simple and brilliant demonstration by Otto Loewi, a German-born pharmacologist. There is an interesting story about how Otto Lewy conceived of this now famous experiment. On the Easter night of 1921, Lewy had a dream about a possible experimental setup that can resolve the question of neurotransmission by chemical means. He got up in the middle of the night and jotted down something in a notebook. The next morning, he looked up what he wrote and found that it is completely illegible. The following night, apparently, he had the same dream. This time around, he woke up and wrote it down in detail with great care. The experiment consists of two beating frog hearts kept in ringer solution. One of them had a nerve called the vagus nerve connected to it. Stimulation of this nerve is known to slow down the beating of the heart. When the vagus nerve is stimulated, the heart connected to it slowed down. Then the experimenter took the liquid surrounding the stimulated heart and injected into the second heart. To the experimenter's delight, the second heart now slowed down. The only explanation to this effect is as follows. When the vagus nerve of the first heart was stimulated, the nerve endings have released some chemical whose action on the first heart was to slow it down. When the liquid from the first heart is transferred, the chemical contained in the liquid acted on the second heart also, slowing it down. This is the first direct demonstration that the signal released by neurons is a chemical substance. Further work by others like John Eccles and Henry Dale had established clearly the idea that neurotransmission is mediated predominantly by chemicals. Such synapses are known as chemical synapses. However, there are some synapses where neurons interact electrically. Such synapses are known as electrical synapses. So far, we have seen that neurons are electrically active entities. They produce these sharp voltage spikes called action potentials. They signal to each other by exchange of chemicals. But how do we use this knowledge to answer the original question that we started with? How does the brain work? How does the brain enable us to see the world, to see the sounds of to listen to the sounds of the world, to feel the world, to manipulate the world with our hands, or to navigate the world with our feet? How do we think, love, and feel empathy towards others? How do I realize that just as I have a sense of self inside me, the feeling that I am a person, others too have their own distinct sense of self? That is a big question. Actually, a lot of big questions tightly packed together. We still do not have answers to most of them. Now, let us get to the story of how people began to understand brain function. Some of our earliest faltering steps towards understanding brain function were actually quite silly, but they pointed in the right direction. One such an attempt was the science of phrenology. The word phrenology means the study of the mind in Greek. It was developed by the German physician named Franz Gall in 1796. It was hugely popular in the 19th century with a considerable fan following in the intellectual community. There was even a center for phrenology in Edinburgh in the UK in the early 1800s. Phrenologists, as the practitioners of this dubious science called themselves, proclaimed that they can determine a person's character simply by feeling around the person's head. The shape of the head is thought to be some sort of a picture of their soul. Their reasoning goes as follows. The brain is an organ of the mind. The mind is composed of multiple distinct faculties. Each faculty has a separate organ, a corresponding center in the brain. The size of that organ or center is a measure of the strength of the corresponding faculty. So, the sizes of various organs determine the shape of the brain, and the shape of the brain determines the shape of the skull. Therefore, the surface of the skull is an accurate index of psychological aptitudes and tendencies. Phrenologists would publish speculative, unsubstantiated maps of the brain that look like those seen in the figure. Although phrenology enjoyed the following of a lot of uncritical lay people, there were some who were deeply dissatisfied about it. One of them was none other than Napoleon Bonaparte, the two-time emperor of France. Napoleon was generally remembered for his military campaigns, but he was also a great proponent of science. Unhappy about the unscientific nature of phrenology, Napoleon asked the Academy of Sciences of Paris to look into the matter from a scientific point of view. The Academy asked Jean-Pierre Flourens, a physiologist, to take up the task. Florence set out to investigate the basic premise of phrenology without going into the details. Are brain functions so precisely localized as depicted in the maps of phrenology? Florence asked. He investigated this question using the experimental methods of modern science. He had taken experimental animals like rabbits and pigeons and removed various parts of their brain to see if elimination of a specific part of the brain produced a specific change in their behavior. Florence did not find any specific localization of brain function. He expressed his findings as follows. All perceptions, all volitions occupy the same seat in these cerebral organs. The faculty of perceiving, of conceiving or willing merely constitutes therefore a faculty which is essentially one. This work and similar studies performed by others led to the so called aggregate field view of the brain, which states that brain functions cannot be localized to distinct regions of the brain, that brain acts as an aggregate or a whole. But the work done by British neurologist Hughlings Jackson had seriously countered that aggregate field view. Jackson studied the effects of focal epilepsy, in which the patients exhibit a characteristic type of convulsion. These convulsions typically begin from one part of the body like the hands and gradually spread to the neighboring regions like the forearm and the shoulder. Jackson had an insight studying the underlying pattern of these convulsions. Perhaps there are specific brain regions controlling different body parts. Abnormal electrical activity probably begins first in one of them, say the finger region, then it spreads like a forest fire to the neighboring regions like the forearm region. From there it spreads to the shoulder region and so on. Thus, Jackson's studies supported the localization view of the brain function, which held that specific functions are controlled by specific brain regions. Jackson's description highlighted another aspect of how brain functions are organized. Nearby body parts are controlled by nearby regions in the brain, and that means there must be some sort of maps of the body in the brain. These insights were quite true and confirmed by subsequent studies. Noted among them were the pioneering experiments by Wilder Penfield. Penfield was a Canadian American neurosurgeon who pioneered a wide variety of surgical techniques. He was particularly known for the technique he developed along with a colleague named Herbert Jasper for treating epilepsy. Epilepsy is a brain disorder that is characterized by abnormal electrical activity of the neurons, which starts from one region and spreads over a wider region of the brain. The site from which such activity originates is called the epileptic focus, which is usually inside the temporal lobe. Penfield's strategy was to identify the epileptic focus and surgically destroy it. However, there is a difficulty in identifying the epileptic focus. In appearance, it looks just like the normal tissue. The only way to identify it is to stimulate it electrically and see if it triggers an epileptic seizure. Thus began Penfield's legendary brain stimulation experiments. Penfield would stimulate various sites in the brain and ask the patient who happens to be awake what she or he felt. When certain areas in the temporal lobe are stimulated, the patients reported that they heard sounds or recalled words they heard long ago. When sites in the occipital lobe are stimulated, they saw flashes of light. When points in the somatosensory area are stimulated, they felt as if someone was touching them. The somatosensory area is the stretch of this brain surface present right behind the long vertical groove called the central sulcus. An entire map of the body was discovered embedded in the somatosensory cortex. A similar map was also found in the motor cortex. On further exploration, it was found that these maps were not drawn to scale. For example, there is an abnormally large space in the somatosensory area allotted to lips and mouth region. Hands also have a large allocation. The trunk is quite tiny. These kinds of maps have been embodied as quaint three-dimensional icons called homunculi. Homunculus simply means the little man. These idols conveyed an oversimplified and somewhat misleading idea that there is a little man who is sitting inside the brain, receiving all our sensations and controlling the bigger man, which is the body. Thus, Penfield's classic studies seem to strongly support the localization view of the brain. More support came from patients with aphasias, a general term indicating difficulties in speaking. In one form of aphasia, named the Broca's aphasia after its discoverer, who is a French neurologist, the patient had difficulty forming complete sentences. For example, a patient who wanted to say that he has a smart son who goes to university might end up saying something like son, university, smart, boy. In extreme cases, a patient might be able to utter only a single word. One of Broca's patients was named Tan because that was the only word he could utter. But Broca's patients could understand language. Their problem lies in expressing them in speech. On postmortem, Broca discovered a lesion in a certain region in the frontal lobe of the brain of his patients. A different kind of aphasia was discovered by Karl Wernicke, a German physician. Wernicke's patients could speak, often quite fluently, but the sentences did not make much sense. For example, unlike Broca's patients, these patients could speak but could not understand speech, even if it is their own speech. I called my mother on the television and did not understand the door. It was to breakfast, but they came from far and near. My mother is too old for me to be young. This is an example from the speech of a Wernicke's patient. Wernicke's patient was found to have a lesion in the temporal lobe, an area that is now named after its discoverer. A third form of aphasia, called the conduction aphasia, which in a sense connects the two previous forms, was also discovered. Patients with conduction aphasia can utter sentences, can understand sentences, but cannot repeat what is said to them. In these patients, it appears that the part of the brain that understands the language, that is the Wernicke's area, is not able to communicate it to the part of the brain that produces speech, that is the Broca's area. Indeed, that turned out to be the case. It was found that a bundle of nerve fibers that connect the Wernicke's area with the Broca's area, known as the arcuate fasciculus, is damaged in these patients. These findings of aphasic studies inspired Karl Wernicke to propose a comprehensive theory of brain function that sought to reconcile the long-standing debate about the nature of brain functions. Which view of the brain is correct, the localization view or the aggregate field view? Wernicke proposed a synthesis of the two rival views. He said both views are true. It depends on the scale at which we study brain function. Let us consider, for example, the general problem of how brain conducts language functions. Language is understood in the Wernicke's area and expressed as speech in the Broca's area. In this sense, the localization view is indeed correct, but if we consider the general language for operations of the brain, several other brain areas come into the picture in addition to Broca's and Wernicke's areas. For example, if you are reading from a text, your visual areas must be involved, or if you are listening to somebody speaking, your auditory areas must be involved. Wernicke coined the expression parallel and distributed processing to describe what happens in the brain. Simple primitive functions are localized, but more complex functions are distributed over multiple brain regions that work in parallel. A similar synthesis has been arrived at by neuroscientists like Ramon E. Cajal or Charles Sherrington, who developed a view of the brain function called cellular connectionism. Sensory information entering the brain is processed by the brain as it flows over the incredibly large number of connections, and the result of such processing is expressed as motor output by our hands, legs, or speech. This connectionist view of the brain had succeeded in explaining a large number of neural phenomena and had led to the so-called connectionist revolution. Neuroscience today is experiencing an explosive growth. We have come a long way since the days of the pioneers like Ramon E. Cajal or Karl Wernicke or Charles Sherrington. Modern experimental methods give us data at an unprecedented scale and depth about various aspects of the brain. Hodgkin and Huxley had used the thick axon of a giant squid due to limitations posed by the blunt electrodes that they had. But today we can record from single neurons that are only a few microns in size. We can measure currents flowing through a single ion channels using what is known as the patch clamp technique. There are bold endeavors to map the connections of the entire human brain called the connectome using modern neuroimaging techniques. There are ambitious attempts like the Blue Brain project to develop computational models of the entire mammalian brains. All this immense activity taking place world over generates a massive amount of data and knowledge. It is not possible to convey all that in a single short course like this, but we will try to transmit the excitement. We will try to convey the key underlying principles of brain function in simple terms. You do not have to be a student of biology to understand neuroscience. You may be an engineer or a student of medicine or physics or chemistry. There is surely a path that leads you from where you are to neuroscience. Modern brain science is extremely interdisciplinary and has something to offer to every one of us. So, let us put on our thinking caps and get ready for an exciting journey. Thank you.